Hello, most of you know me. I'm Seth Wanamueller. I'm here to introduce our speaker today, Dr. Diana Lopez Arias. She joined the University of Minnesota 3 years ago now as a postdoctoral associate in Dr. Stan Hokanson's lab, where she's worked on roses and has helped many people with her expertise in quantitative genetics. She comes to us from, I almost said it, Angers, France, where she did her research at the Research Institute of Horticulture and Seeds. Her research focused on the phenotypic and genetic characterization of the partial resistance to black spot disease in a member of the Rosa visseriana species, and she's continued some of that research here at the University of Minnesota. And that's what she's going to be talking about today, and we're lucky to have her because she's only here for another 2 weeks before she moves back to France. And I'm happy to say that as of yesterday was offered a position at Nestle. Okay, I just wanted to make sure that it was okay that I shared it. Where she's going to be breeding coffee. I'll let you take it over.
Thank you. So this is some sort of a presentation to show you what I've been doing those past 3 days. 3 days, no, 3 years. It seems like 3 days, but no, 3 years. So we'll be talking about molecular breeding for rose black spot resistance, and so our current research and what that has unlocked for us in the future, mainly us, Seth. So before we start diving into this, no, not working. Okay. Should I connect it somehow? I mean, I can see my name. Let me just give you a second. It worked earlier. Oh, good. Alrighty, it was asleep. So I would like to introduce what rose black spot disease is, and then talk about the project and what we do at the Hokanson lab. And then I would like to use 2 case studies of two of our projects that cover the loci mapping part of our research, and then the genetic marker development. So let's talk about rose black spot. Here you have Rosa chinensis all blush. That is a beautiful rose that's known for its fragrance. Very old rose that people love, but actually it is extremely susceptible to rose black spot. So this picture was taken end of June, and you can see that the disease is starting to progress. The most common symptoms for rose black spot are these lesions that are on the upper side of the leaves. They have a particularity of being French margin because the fungus colonizes the leaves and shoots the hyphae, and so that's what you see here at the edge of the lesion. The infection is quickly followed by chlorosis, and premature defoliation can be observed. So here you have a picture of, again, Rosa chinensis all blush, and it was taken beginning of end of July, so about a month later. And you can see that all the leaves have been dropped, and the new foliage is starting to be infected. So this is definitely not what gardeners want in their gardens, and it definitely decreases the quality of the plant. In very susceptible cases, this premature defoliation can lead to the plant's death. And so this disease is caused by a fungus called Diplocarbon rosae. Maybe some of you have heard the name Marcellina rosae for its asexual stage. It is a disease that is found in epidemic proportions, since the fungus is able to complete a whole infectious cycle within 7 to 10 days. So it can do a lot of these cycles throughout the growing season of the rose, and can quickly infect all the parts of the plant. So it is also propagated by water splashes. So its incidence increases at each rain. And also if gardeners use overhead watering. Also, it's a fungus that can overwinter in dormant stems, prickles, and fallen leaves, which makes it very difficult to get rid of. So once the plant is infected, it will just get the disease once again next year. So this fungus is a hemibiotroph, which means that at the beginning of its cycle, it has a biotrophic stage where it feeds from alive cells using this bottle-shaped structures called hostoria. And very quickly, about 5 to 6 days, turns into a necrotroph and is able to feed from dead cells. So it's very adapted to roses. So far, 14 races have been described, and the most efficient way to manage the disease is actually fungicides. But more and more genetic resistance is considered, and breeders have been focusing their efforts on developing cultivars that are disease resistant. So for rose blackspot, we divide the resistance into 2 types. Complete resistance, that is the main focus here at the University of Minnesota and University of Wisconsin River Falls. And then partial resistance that is being studied in France and also at Texas A&M. So complete resistance yields no visible symptoms, and that is because the pathogen is quickly arrested at the penetration points or its growth is stopped. So you have no sporulation that happens and barely no colonization under the cuticle. So some resistance don't allow the spores to germinate. Others just stop the pathogen at the entrance of the cuticle. And there are resistance that are associated with either a localized hypersensitive reaction, so cell death around penetration point, and then others that have more of an extended HR, but no other fungal structures can be found under the cuticle. So this complete resistance has been shown to be conferred by major resistant loci, and is also race specific. So now knowing that disease resistance is so important, I'd like to separate our research into 3 main categories. So first, the segregation population development that is necessary for loci mapping. And then once loci have been identified, we are able to develop markers for breeding. So for the segregating population development, well, most of you here do breeding, so I'm going to go very quickly with this. We cross a resistant seedling with a resistant parent with a susceptible one, and we use these populations for loci mapping, but also to identify germplasm that can be used by breeders later on. And so some roses are selected for germplasm release. And here, for example, you have seedlings that were selected from a cross between Mordenblush that looks like this, has very pale, beautiful, fragrant roses, and then George Vancouver that has more of a pink red. And so these seedlings here all share the same resistance than the resistant parent. But I want to point out, for example, this one, it has the features of the susceptible parent, but carries the resistance. And so these germplasm release can be a way for breeders to quickly use these seedlings as starting material for their breeding programs. And so these populations are mainly used for loci mapping. And so how do we do this? So we need 2 types of data sets, the phenotyping data set and the genetic map. For phenotyping, we use laboratory assays where we use detached leaf assays and we infect seedlings, all the seedlings of a population, as well as the parents with different races of Diplocapron rosae. And to do that, we use fungus that is actively growing on leaves. And so here in white, you can see the sporulation happening, and we just use those leaves to obtain the new spores from the acervuli right here. So this is under the microscope an acervuli that has erupted. You can see the cuticle just opening here, and it holds hundreds of thousands of spores. So we use these infected leaves that we place into tubes with distilled water. We set the concentration at 75 spores per milliliters and deposit that inoculum with droplets on the top, on the upper side of the leaves. We leave those droplets for about 2 to 3 days because that's how much the fungus needs to be able to successfully infect the plants, the leaves. And then about 7 to 10 days, you start seeing the lesions forming. Some genotypes that are so susceptible can develop these lesions after 4 days. But our phenotyping part takes place after 14 days to leave the chance of some slower growth. So the phenotyping is fairly simple. We just look at whether or not a rose is infected and produce acervuli, and if it has, if it doesn't have any infection, then we can see some hypersensitive reactions. So we score all of these seedlings and determine whether they are resistant or susceptible. This binary scale has been really useful to detect a lot of major loci, and so I'll be talking about that a little bit later. And so for the genetic map, I just want to step back a little bit and talk about the rose genetics. So roses have a set of 7 chromosomes, and the ploidy ranges from the diploids to undecaploids. So thankfully, most of the cultivars are diploid and tetraploid. In our case, the garden roses that we work with are tetraploid. So this is how it looks for people that don't know much about ploidy. So it has 4 sets of homologous chromosomes, and so a total of 28 homologous chromosomes. So for the genotyping, for diploids, what you're most likely used to is having either SNPs that are homozygous AA, SNPs that are homozygous BB, and then in the middle you have the heterozygous SNPs. But for tetraploid SNP genotyping, you will still have your homozygous A and then your homozygous B. But for the heterozygous, you can have dosage of that B allele that can differ. So you have SNPs that can be in simplex, so 1 dose, 2 doses, 3 doses. And so these are the ones that are used for the mapping studies. So in roses, we have an AXIM array that we use that has 68,000 SNPs that were developed using a large collection of roses that were for garden use and then for cut flower use. I'm just going to briefly talk about the main steps for genetic map construction for a tetraploid rose. So there are many packages that you can use, but we use polymap R since it has the lowest computational power needs, and it's the most straightforward way to get a linkage map of high quality. So the first step is to use the simplex by nudiplex markers. So either simplex on one parent and nudiplex on the other one and vice versa. And we use these markers to group all of the markers into 28 plus clusters or homologs. So I say 28 plus because most of the time, some of the homologs are broken down in different clusters, and afterwards you will be able to combine them into actual linkage, homologous linkage groups. So that's the first step. Then after you have identified those different clusters, you can group them into different linkage groups. And so you're looking for getting 7 linkage group, and so you have to kind of play around with the lot and try to see what the most efficient grouping is. The second step is to use the simplex by simplex markers that allow you to actually find groups of markers that belong to a homologous chromosome. So for example, here for linkage group 1, you have the 4 expected homologs that were constructed. Before getting to that, you had, for example, 2 clusters for homolog 1 that were separated. So our work was to actually just bridge those clusters into a single homolog. And then the first step, once you have that backbone of simplex by nuliplex and simplex by simplex markers, you can just add up all the remaining marker classes and start ordering your markers along the linkage group. And so this is the example of a linkage map that just came out of these steps. You still have a lot of work to do by removing some of the markers that have high stress, but you can get really good clustering and then positioning of those markers. So once you have the genetic map, you can use the phenotypic data, so that binary phenotypic data, to map the resistance onto the different linkage groups. And for that, we use a software called polyqtlr. You have other types that are available, but this one is, again, the most straightforward one for us. So using a similar approach, our colleagues in Germany have identified RDR1 and RDR2 from the Rosa multiflora background, and that was localized on chromosome 1. Actually, RDR1 is the first rose black spot resistant gene that had been cloned. And that was started in the year 2000, so a long time ago. So here at the University of Minnesota, we mainly the people before me have identified 2 loci that were associated with complete resistance. So one from George Vancouver that is located on chromosome 6 and that we call RDR3. One from Brad Eyes that was located on chromosome 5 that we call RDR4. And then since I started working here, we have identified RDR5 on chromosome 7 from Rambling Red. And then with the work of Emily, we have identified RDR6 from G168 that is a descendant from Baby Love. And so that one was located on chromosome 5, but at the very beginning, which shows that it is different from RDR4. So what is interesting to see is that these different cultivars have very different spectrum of resistance. And so that tells us that complete resistance is, yes, race specific, but some of them have different spectrum of resistance. So for instance, RDR4 gives resistance to 12 races out of 13. RDR6 give resistance to 11 races out of 13, and then 3 and 5 give resistance to 9 races out of 13. But what is interesting is that they give resistance to very different races compared one from another. And so knowing that, our aim would be to pyramid those genes in order to have a more durable and efficient resistance. So for instance, if we stack RDR3, 4, and 5, we are able to stop all the known races of Diplocarbon rosae. And that is the aim and maybe future projects with Seth that will be happening. So then the last area of focus, and that's something that I had it was really important for me to just make that knowledge here available for breeders, because they always say, yes, we like that you're working on rose black spot, but what does that change for us? We know where it's located, but we can't do it. We can't do anything. We don't have enough money to develop our own markers. So when I arrived here, I wanted to make that knowledge available. And so we started a whole new pen of research that was the marker development for those breeders. And so our idea was to develop a breeder's toolkit that will help them to breed more efficiently for rose black spot resistance. And that toolkit comprised genetic markers and resistance information. So the genetic markers are important and interesting for the breeders that have enough money to run those markers, and they have expressed interest on testing those markers on their germplasm. And so we have been working on developing those markers for them. So far, the most interesting technology that we can use for this type of markers are CASP or PACE assays. They are very affordable and easy to use. And breeders can go to a third-party company and just run those through them very, very easily and at lower cost. Then for breeders that do not have that kind of money, but that are still interested in knowing what is out there in their germplasm, we have phenotyped about 300 cultivars in breeding selection from different sources and different breeding programs. And so the knowledge of this resistant information, so if it gives resistance to many races or just a few, can help breeders make their decisions into, okay, I'm going to be using this cultivar in my breeding program combined with this one, so I can have a whole spectrum of resistance. So this is actually quite interesting for the small hybridizers, and we hope that they can use that information later on. And so now I would like to just talk about two main projects that were very successful, one for loci mapping and the other one for marker development. Let's talk about rambling red. So we developed a population that we cross the susceptible cultivar Mordan blush with rambling red. And so this is our population in the greenhouse. So we keep it until we're done with the detached leaf assays. And so we inoculated the whole population and the two parents with race 5, 8, 9, and 11. See, here is an example of the inoculation box of Mordan blush after 7 days with race 9. So after inoculating all this population, what we found out was that Mordan blush, as expected, had a complete infection, was completely infected by race 5, 8, 9, and 11. You can see strong development of the pathogen. Conidia is released, acerbola is produced, strong hyphae network. So that was what we expected. On the other hand, rambling red was extremely resistant to race 11. No infections, no hypersensitive reaction was observed, which was very typical of what we are used to with this complete type of resistance. However, when we infected rambling red with race 5, we saw some sort of partial infection of that cultivar when inoculated with race 5. We saw not as much infection when it is fully susceptible. Some HR associated with a very slight hyphae network, developing sometimes very sparse acerbola and some sporulation. So that was kind of interesting. So for the population, how those phenotyping results look like. So for race 8, 9, and 11, we found exactly the same results. 62 of the seedlings were resistant and 38 were susceptible. This was a surprise because for RDR3 and 4, we had a 50-50 1-to-1 segregation ratio, which is something expected when you have a single major loci. But in this case, we found a 2-to-1 segregation ratio. And for race 5, and that was the most surprising, was that you had even more susceptible seedlings to race 5 than for race 8, 9, and 11. And when we looked a little closer, we saw that you still have those 41 that were actually resistant to all races, 5, 8, 9, and 11. But within these 58, 37 of them had a susceptibility to all races, but these 21 here were only susceptible to race 5. So what that tells us is that the mechanisms or the loci behind the resistance observed in these two races are different. Most likely, there is a second loci or something going on when rambling red try to resist to race 5. So for the linkage map, we were able to build an integrated and parental linkage map with about 6,500 markers. The total length was very similar to what was observed in the literature, but our map was definitely of higher density, and we were able to retrieve all the 28 homologs, which sometimes is not the case because of that use of the simplex markers. Sometimes you don't have enough simplex markers to identify one of the homologs, and so that can happen. So after getting the phenotypic data and the genetic map, we're able to identify one major loci that explained between 80 to 96% of the variation, and that was located on linkage group 7. So I'm going to talk about this one first. For the data sets for race 5 and race 8 and 9 and 11, the loci that we identified colocalized to the exact same region. And what we found is that homolog 7 of rambling red, so homolog 3 of rambling red, is the one that brings the resistance. You can see here that seedlings that inherit this region tend to have lower disease scores than the ones that inherit other homologs. And then, and that was our biggest surprise, was that when we fixed the effect of this specific loci, we're able to identify this loci here on chromosome 4. So this one was only found for the phenotypic data for race 5, no surprise. But what was interesting was that homolog 6 of rambling red was the one that controlled that phenotype observed. But what we saw is that instead of bringing resistance, it was bringing susceptibility. And that was something that really surprised us. So because of those those results, that was what I was supposed to be working on, just finding RDR5, publishing that, it will be done. But seeing these results with race 5, we had to push it a little further. And so because it was not a susceptible versus resistant type of trait, I decided to start working on using that a scoring scale that would allow us to see more quantitative or at least semi-quantitative response to the disease. And so I used this 5 scoring scale. We re-inoculated the population and then scored them with this, but also measured lesion length and the absence presence of aceruli. But I'm just going to talk about the results that we found for that scoring scale. To go back to what we used, everything that was scored 0 and 1 was previously considered resistant, and then everything that was scored 2 and above was considered susceptible. So these were the results that we obtained when we re-inoculated the population with race 5 and race 11. So talking about race 5, it was very straightforward. Seedlings that inherited RDR5, which is the resistant, the loci that gives the resistance, but didn't inherit RDS1 or inherit RDS1 too, were completely resistant. The ones that did not inherit RDR5 were completely susceptible. So that was pretty straightforward, and this is how it looks. So RDR5 that gives resistance was located on chromosome 7. RDS1 that gives susceptibility was on chromosomes 4. And so when seedlings did not inherit RDR5, no matter if they do inherit RDS1, they were susceptible. If they inherited RDR5, they were resistant. So that was something that we are used to, one gene controlling the resistance, and that's it. But then when we looked at the population being challenged with race 5, we still saw that when you only inherit RDR5, you are completely resistant. If you don't inherit RDR5, completely susceptible. But what was interesting was that if you inherit RDR5 alongside RDS1, the seedlings started showing some partial resistance, partial susceptibility. At least they were more infected. You had some development of the hyphae, sparse correlation, but it did not look like it was completely susceptible. So it seems that if you only inherit RDR5, you're completely resistant. But if you have that allele on RDS1 loci, then the effect of RDR5 is no longer valid, and it doesn't give the resistance that we can observe when it's inherited alone. And so that tells us that RDS1 does give some susceptibility, and that was some susceptibility to specifically race 5. And so RDR5 gives resistance to all races that we tested, so race 5, 8, 9, and 11, but that RDS1 has a race specificity, and it does give some susceptibility. So that was something unexpected. So I wanted to show that to students that sometimes you have a hypothesis and you're like, okay, this is going to be very easy, but then you see that other things can actually come up. This is the first time that a susceptibility loci was discovered for rose black spot resistance. And so it's good news. We have RDR5. We can tell breeders to use that. But bad news, rambling red that has a true red does have in its background some susceptibility that needs to be taken into account when using that specific cultivar in breeding programs. So now let's move on and talk about marker development. So because we don't do and we don't have a breeding program in our lab, we wanted to develop markers that breeders can use in different genetic backgrounds. And so Platten in 2019 actually said that, yes, most breeders are trying to integrate mass, so marker-assisted selection in their programs, and they are looking for marker traits associations that are robust and that they can actually use with different elite materials and breeding programs. But the case right now is that markers that you found and that are developed in one specific genetic background are actually very difficult to use when you move outside of that specific background. And so there is a low transferability that is problematic. And because we needed this technology to be out and available and useful for all the breeders that we work with, we wanted to develop a strategy so that we can develop reliable markers for them to use. And so this is a strategy that we use. So first, we decide to, of course, go the closest we can to the causal gene of RDR loci, and then develop what we call cultivar-specific primers to be able to avoid some primer failure or issues when the PCR is happening. And so what we try to avoid here is SNPs in the primer region. And so by developing markers that are in with the sequence of that specific cultivar, we can easily circumvent that issue. And then we wanted to have those markers validated on distant material so that we are sure that those markers work, even if a breeder wants to use it in very distant cultivar. And so I'm going to present a case study of RDR3 because it was our crash test to see if that strategy worked. And so it had been already localized to chromosome 6. Some SSR markers were developed that were pretty closely linked to the loci. But we know that ISSR markers are not that easy to transfer between labs and between germplasm. And also, there are less and less companies that are willing to genotype those markers. So we needed to move forward and use another type of marker. Also, in that publication, they were able to go up and say for sure that Champlain was the donor of RDR3. But as long as when they moved up in the pedigree, they kind of lost the linkage with the trait. So our aim was to develop a marker that actually could be used even in distant related roses. So because at that time we didn't have money and nobody really believed that we could actually do that, we used only 3 resistant cultivars that we knew carried RDR3. So George Vancouver, Folksinger, that is somewhere in the pedigree, and then a seedling that was resistant. On the other hand, we used 3 susceptible cultivars that do not carry RDR3, Morden blush, that is the susceptible parent of the population, a seedling that was susceptible, and then a random cultivar that had no link with neither Morden blush or George Vancouver, just so that we covered our basis of detecting SNPs that are not in the germplasm. So again, we didn't have much money, so we had to kind of work around that, and we used amplicon sequencing. We targeted about 400 base pair of that 2 megabase region, where we selected 11 NBSLR genes that we sequenced. And those sequences were then used to do some very basic variant discovery. And the aim was to find SNPs that were exactly the same, that had exactly the same base for the resistant group, but that was different from the susceptible group. And so that allowed us to identify about 2,000 SNPs in both of the genomes. So these genomes are Rosa chinensis genomes. They come from the same plant, but at different recombination events. So they're very similar. But we still use them both just to see if we had a difference. And through different steps of harsh filtering, we so we only focus on the ones that were on simplex configuration, so simplex by nuliplex, because we knew that the gene was on simplex configuration anyways. And then we selected the ones that had the least amount of missing data, very deep sequencing, and the other criteria. So we were able to lower down our SNP selection to 78 and 82 SNPs. Actually, 69 of them were common between these 2, which is reassuring because they're supposed to be the same genome. But 9 were specifically found in one genome and 13 on the other one. So those 107 SNPs were then again filtered, and to avoid any issues of SNPs in the marker in the primary regions, we removed SNPs that were overlapping with 75 base pair or less, and only kept the ones that were further apart enough so that we wouldn't have any issues with those SNPs in the primary region. We selected 48 of these markers and test them back to the segregating population. And usually that's where most of the publications stop when they develop markers. So as you can see, those 48 markers, we found 3 that had primer design failure. They were not able to design a primer for those 3. 7 of them were actually homozygous in the population, which was interesting because by looking at the parents and one seedling, they were heterozygous, but when you look at the whole population, they were actually just homozygous. And then 21 of them did not clearly differentiate resistance versus susceptible. Most of them actually in this category had high missing data rate, so these were not considered. And then we had 17 that we're actually able to call resistance versus susceptible correctly at 99% confidence. And so we said not between 95 and 99% of confidence. And so we selected 11 of them to be tested on the cultivars that were already confirmed to have RDR3 with the SSR markers. And they worked at 100% call rate, which makes sense because you see the SSR here, the SNPs that we found were within that region. So it makes sense that it worked perfectly. So then our next step was to use those 11 markers and validate them on close relative. So do you remember the pedigree? We used that pedigree, but also extend the collection beyond the close relatives of George and Cooper. And that helped us identify that these SNPs here started losing marker trait association the further we went in the pedigree and the further we went from George and Cooper. And these three here were actually the ones that work for all the 208 markers that 208 cultivars that we tested. So these are the candidates that we kept because we know that they can successfully differentiate resistance versus susceptible, and that it can work for very unrelated SNPs cultivars. Something interesting that we also noticed, so in all this, because we were focusing on simplex by newlyplex markers, it was basically just working like we're working for duplex for diploid cultivars. But let's not forget that these are polyploid roses, which we can see here that we were expecting, for example, the SNP number 16 to be either homozygous for allele A and then heterozygous for allele A1. But some of the cultivars actually started showing that they could also be homozygous for allele A1. And maybe we can even pick up some dosage within the panels that we use. The machine does not allow for polyploidy so far, so that's why we had to remove those in order to be able to see the different colors. But we can definitely see that this group of seedlings, or this group of roses, have the expected dosage of simplex by newlyplex, but these two cultivars here seem to have maybe a different dosage than what we were expected. And these were resistant, so as well as this one. So it is interesting to see that having at least one homolog that carried a favorable allele is enough, and that our assays can actually maybe detect even dosages among these homologs. Another nice news was that we were able to actually identify the wild species that from which RDR3 originated from. I'm not allowed to talk about it just yet because we want to keep the surprise for our collaborators, but the use of these three markers really told us that, okay, this specific wild species is the one from which that resistance originated from. So that was really encouraging. Beyond that, because we were able to narrow down the region to these three markers, we went from a 2 megabase region to about 150 base pair, which reduced considerably the candidate gene list to about 12 candidate genes, with among them 2 that are NBSLR and 2 that are ROP interactive types. So for further work, and I'm looking at Seth, if we want to clone that gene, this reduction of the interval is actually really promising. So the success of this project for RDR3, even though we had a lot of strong hypotheses that had to be drawn, such as using only 3 three cultivars that were sequenced, or using an unrelated genome from a very different species, but also only sequencing NBSLR genes. We showed that even with these strong hypotheses that we did at the beginning, we were able to successfully develop pace markers that can be used and transferred from different genetic backgrounds. So that finally got the attention of the industry, of the university, and we were able to found even more projects for market development for all the markets that are developed here at the University of Minnesota with the University of Wisconsin River Falls. And so we started the development of a marker for RDR5 thanks to the Glockner Foundation grant, and we should be getting the results fairly soon. We have learned from our mistakes, and then we are now developing this marker using GBTS, so genotype by target sequencing. So we target RDR5 region, but we sequence the population and some of our validation cultivars. So the detection of the SNPs that we will be getting is much more reliable. And we'll have less homozygous and loss of marker trait association. We are currently also working on developing a marker for RGR6. And so that is mainly done at the University of Wisconsin River Falls right now. And then we have a project with Seth to do the market development for RGR4. So this is, if you remember, the one that gives resistance to 12 races out of 13. So it's a really important loci for us, and also because bright eyes have been used extensively in breeding programs. And so we're putting a lot of effort on developing a good, reliable marker, and even try to just land on the gene directly when we develop that marker. And so for that, we're using a combination of fine mapping, genome development, transcriptomic, to be able to really fine map and develop markers that are very close or on the gene. So hopefully we'll get the HRI grant to be working on this, and this is a very exciting work because it'll involve collaborations with friends and other actors. So just to summarize all this, gross black spot resistance has two types of resistance that have been extensively studied in different parts of the world. Here we have detected loci that are associated with complete resistance. It is, even if breast specific, it is broad spectrum and it yields no symptoms. On the other hand, you have the partial resistance that I worked during my PhD, and loci have been detected for this type of resistance. It is considered more durable in some ways. I know that there is a whole discussion of like, is it actually? Some partial resistance are actually race specific and not as broad spectrum. So a further study needs to be done to understand if it's actually more durable. In addition, when I do a talk about this with gardeners, and I ask them, so what do you prefer? Would you want to have a rose that is completely free of disease, or would you accept having some spots in your roses? The answer is almost unanimous. No, I don't want to see any black spot. So there is still a lot of work to do to make them understand that actually partial resistance is something that could be better for them, because we all know that complete resistance based on major genes can be very quickly overcome by the pathogen. And so by working together with those labs, we can try to pyramid these genes, but also have this partial resistance that could be the last layer to protect the rose. But because it is an arms race, and like I told you, the pathogen is constantly evolving and trying to break the resistance, there are new races that can start infecting rose black spot. And so that's why our work is so important And all the different steps are very, very necessary, especially developing segregating population, because this is the beginning of all the steps that we can provide, so that we can provide breeders with markers. And so all this work has been funded by Rosebreed 2 and the Sustainable Rose Scree Grant. There's a lot of people, and of course, we have to say a big thank you to the American Rose Society that believed in us and funded the RDR3 work so that we could finish the validation. And so thanks to that help, we were able to show that, yes, we can develop markers and that are reliable. So all this work is in conjunction with our team in Wisconsin River Falls. They pull up most of the work for the phenotyping and the genotyping of those markers. And we here work on the development of the markers itself with the bioinformatics, and we keep some of the collections here too. We have collaborators all throughout the United States and also in France that also help us and will help us move forward with the marker development. So if you have any questions, just feel free to ask them.